Hey everybody, it's Mr. Caudell. We are inside of chapter 9. Specifically, we started chapter 9 with 9.1, and we're about halfway through 9.1. In the 10th edition of Campbell, we're going to go over today, starting at a page 165. We're going to talk about NAD and FAD. We'll get to the big, broad overview of how ATP is made by the breaking down of food. And then we'll get into more specifics about glycolysis, and then we're going to talk about the link reaction and then the citric acid cycle, and that's it. So we're probably not going to get all the way through 9.3, which is the citric acid cycle, but I would say you should have a pretty good idea of what the citric acid cycle is by the end of this lecture. Make sure that you have this. This is my simplified graphic of the steps of cellular respiration. It has a bat on the front of it. It's from an old textbook. I don't even have a copy of it anymore, but I just like it, and I like how succinct it is. Anytime you see these big cartoons that don't come out of your textbook, refer to this. You should have this handout. I do want you to read the whole chapter, but I want you to really focus on this level as far as when you go to memorize everything for the exam, and I'll get to all that detail. Well, let's start out like we normally do with a song.
Tell me what you want, what you really want. I'll tell you what I want, what I really want. I want some energy. I want some energy. If you break down glucose, you get pyruvate. Pyruvate dehydrogenase gives you two simple ways. To the mitochondria is where we'll go. Start the CDC cycle now, we'll take it slow. So acetylCoA plus phosphate, catalyzed by citrates and bases. Citrate is what they make this concentrated reactor makes that one. If you want to make some energy, you gotta go through 8 steps. Need acetylCoA to activate the process. 1 GBP we remove, 2 CO2 to lose. Reducing free ADH and 1 ADH2. What do you think?
Okay, so sorry about that. Certainly you can look up online any cute jingle that will help you with this stuff. I think it's great because it's super complicated and hard for kids, and you can see the coping mechanisms that have been created over the years, and they probably have helped kids who are humming along during the test. So remember, this is all about carrying electrons which come from hydrogen atoms to the end of this process, which is the electron transport chain, and that's where we really make some ATP from ADP plus phosphate. But how do we carry these electrons? You see it right here and here. It says electrons are carried. And the answer is, I alluded to this the last time we spoke, was NAD and FAD. And so NAD is a plus. It's a charged molecule. Same with FAD. But you can see that here, as following glycolysis in the citric acid cycle, NADH and FADH2 account for most of the energy extracted from food. I'm going to show you in a moment where 10% of the energy is made, but let's just focus on the big picture now of getting the hydrogens, stripping them off of this molecule of glucose, and then taking those electrons, which were once part of hydrogens, to the electron transport chain. You can see it here, 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 and here. Just know that this is a big player, and it's an electron carrying molecule. You can see then that there's a couple of ATP made at the end of each glycolysis in the citric acid cycle. That's made through a different process. I'll touch on that in a moment. But about 90% of all ATP is made at the end of the electron transport chain. So what's important really is the FAD and NAD that carry those electrons. You know, who makes 38 ATP? I looked it up even again this morning. Before this, I did this. I heard that one type of seahorse does it. I think in super ultimate conditions, I don't even know what that means. As far as everything's working swimmingly well, maybe that happens sometimes in higher organisms. I just don't know. It's just all over the place every time I look it up. I even read this morning, scientists now believe that we only make like 30 to 32 ATP per glucose that's broken down. So I don't know the answer to this. Just memorize somewhere in between 30 to 38 ATP. As far as how much energy can we extract from this. Well, recall that the hydrogen atom is comprised of two things, an electron and a proton. And we like to draw a proton. We can draw a P down here with a plus next to it, but we are basically going to say that the only thing that's left of a hydrogen atom when an electron gets taken away from it is the proton, and you could draw that ion. If you take the electron away, it's H plus, like we would draw Na plus for sodium ion, or Cl minus. We're just drawing an H, so it's a hydrogen atom, and now it's a plus because it has lost an electron. So fine. Just know that these are the two things that are made when a hydrogen atom breaks down. And so you can see on this picture, there's some complexity here. Way up at the top, you can see that there's two electrons and two hydrogen ions, or protons, those are interchangeable terms, that are liberated when, you can see it, the kind of midway left there where it says from glucose, when two hydrogen atoms are taken off the glucose. Well, here's what NAD looks like. And here's what NAD looks like. You can see that it is a positively charged molecule. You can see there that the N, the nitrogen that exists in that ring structure, kind of off the main structure, has lost an electron. So it has a positive charge. This thing carries electrons to the electron transport chain. FAD probably looks similar. We don't have a picture of it in the textbook. That's fine. Here's kind of the nuts and bolts of what happened. You have the NAD plus. It grabs the two hydrogen atoms from a glucose molecule. Literally, it's going to tear these things off, and I'm about to tear these things off. You can see way up there at the top then that the two electrons are added into the molecule. One of them is going to go to reduce the charge on that nitrogen atom that exists in that ring structure. So now we just have an N, right? We had an N plus, now we have an N because we gave it an electron. And then the other electron plus proton are going to come together to make a hydrogen atom. And you can see that there. That's that red H that's up in the top right-hand corner where it says nicotinamide reduced form. You can see that red H that's off the ring structure. Well, what's left over? We had two electrons, two hydrogen ions, or these protons. Both of these were used, but only one of these were used. And you can see that's why in the far right-hand corner of this picture that there's a hydrogen ion that's left over. I can talk later about where that thing goes. But please press pause if you need to really understand this picture. Why don't you even take a moment? I think it's important to be able to explain things, to show mastery. So go ahead and explain this to your neighbor.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron, a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme, and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
Okay. Understand that there's three major stages here. We've gone over these. We're going to go over them again. We have glycolysis. Glycolysis is going to basically break down this glucose into two molecules called pyruvate. There's some other products in the reaction, and I'll give you some details about where it happens and why it happens. And then the pyruvates then go into the mitochondria. We have then the link reaction, which prepares the molecule for the citric acid cycle. And it's named that because citric acid is kind of the reactant that it binds to, the thing that comes out of the link reaction. I'll show you where that is in a moment. Then you can see here that as a result of both of these reactions, you do get some ATP created in substrate level phosphorylation. All that means is that you're phosphorylating an ADP with an inorganic phosphate to make ATP. It's just putting a phosphate group on something, well, onto ADP. All of a sudden you make ATP and you're like, sweet, that's an energy storage molecule that we could then break down and contract a muscle or beat a heart or whatever, enable eyes to see. So how does this happen? This is only 10% of the ATP that's made in the breakdown of this.
So substrate level phosphorylation, so this is going on here. We have compounds that have phosphates attached. They have to get rid of those phosphates. And phosphates can be added enzymatically to ADP to make ATP. So substrate level phosphorylation is actually a very straightforward thing. You're simply taking a phosphorylated compound. That phosphate on that compound is being added to ATP to make ATP. This is a representation of ATP. It's a phosphorylated ATP, so it's an adenosine diphosphate plus a phosphate, so adenosine triphosphate.
Don't you love the hippie professor with the tie-dye? I bet he had Birkenstocks on. So 10% of ATP is formed in glycolysis in the citric acid cycle by this process. You'll see a paragraph or two in your book on it. It's important, but it's not as important as this big picture. Just understand it. It should be simple. And let me show you where it exists. This is one of the new cartoons from the other book that I want you to focus on. You can see that it happens here and again here and again here. Not at the bottom. The bottom there is representative of the electron transport chain. So we'll get into all this detail. This might look like a crazy picture, but I'll break it all down for you. So 90% of ATP synthesis is made through oxidative phosphorylation, like I said. Let's pause and let's go over what we've just gone over with the NAD, and then talk about the broad picture, because we're about to dive into each of these, starting with glycolysis in more detail. So press pause and recite, like verbally into the air, what you know so far. Well, let's get to glycolysis. Let me show you. It's way up there. This is the bat picture. Way up there at the tippy top, it's kind of a forked image. And basically what we're trying to do here is we're trying to break down glucose and start the process by which it can further be broken down to make ATP. I'm just going to get my hand out here so I can read with you. So I would have someone in the class do this, but let's read at the bottom. Figure 7-2. It says, within the cytoplasm, a 6-carbon glucose molecule is broken down into a pair of 3-carbon pyruvate molecules. Although 4 ATP molecules are generated, 2 ATP molecules are used up in the process, yielding a net energy gain of 2 ATP molecules from each glucose molecule entering glycolysis. Way up there in the top right box on the picture, it says these 2 ATPs are used up in glycolysis when each donates 1 phosphorus group to the sugar molecule. Okay. Down in the bottom, the box, it says a pair of 3-carbon molecules, each containing 2 phosphate groups, donate these phosphate groups to ADP, forming a total of 4 ATPs. And then it looks at the bottom, it says 4 ATPs are formed, 2 are used up, and there's 2 ATP net energy gain. Well, let's pause for a second and let's write some things on the board. So here's what you need to know about glycolysis: the where, the when, the where, the why, and the reactants of the products. We'll start there. So where does it happen? It happens in the cytoplasm. Remember, the cytoplasm is a solution that surrounds all these organelles within the interior of a cell. We've got no oxygen necessary. In fact, they believe that glycolysis, science does, that it's a super ancient process, because we have organisms which have bifurcated in the evolutionary tree of life from higher organisms like, say, humans and elephants, these super simple, like bacterial type organisms that still do this same exact process using the same exact enzymes that we do. So it must have evolved long ago before there was that split in speciation that occurred. And therefore, it must be important and it must work. It must stand up to the test of evolutionary time. All these naturally selected processes trying to knock it down and trying to find a better way when DNA mutates, makes new proteins. And maybe this is a better way to break down food. And it just hasn't happened in a billion years or longer with glycolysis. So it's very good at what it does. I'm going to say, well, there's two ATPs that are made, and then we have these two NAD pluses, which get reduced to NADH. So that's kind of the summary. Understand, I would say that 2-pyruvate are made, and then they're going to go into the middle or the matrix of the mitochondria to be then link reaction. I'll write down here. I'll write down here link, and then there's going to be this. That's supposed to be a circle, a citric acid cycle that occurs from there. So we'll get to that detail in a minute. Let's do this though. We have this glucose molecule. Let me show you, and I would have you do this if you were in class. Let's tear some hydrogens off of this. So I'm going to go back to this picture, and if you follow along in your little cartoon, you're going to see that that among other things, what we're going to try to do is split this in half, essentially. So I'm just going to do that right now, and we need to take off 2 hydrogens. So here's 1 hydrogen that I've taken off. Here's another hydrogen that I've taken off. So all I've taken off is 2 hydrogens. I know here that if glucose is C6H12O6, then by the end of this process, I should have C6H, that's not true, not H10, because every time an NAD gets reduced, it takes 2 hydrogens. So actually this is going to be H8, because there's 2 NADs that are reduced, and each of them took 2 hydrogen atoms in order to do that. So this should be C6H8O6. As far as what I've stripped off this molecule, we know that it's then going to split into 2 things. It should be C3H4O3. So let me get that. So C3. Bear with me. I think this is important to see this. C3. O3H4. Okay. So let's just imagine that this is correct. So we have 3 carbons, 4 hydrogens, 1, 2, 3, 4, the 4 white kind of plastic things here, and then 3 oxygens. So maybe this is a pyruvate, but you're about to learn that it isn't. But it should be. I don't understand why your book is going to tell you that a pyruvate is C3H3O3, but I don't get it. I don't get where the other hydrogen left. How did one more of these hydrogens get knocked off here after what we just talked about? That 2 hydrogens need to be grabbed by each NAD that are going to be reduced. I don't get it. So anyway, so let's bear with it. So let's continue here. So here's the summary of glycolysis. You know, in the bottom banner of the pages in your book on glycolysis, you'll see this long and complicated process here. And it is complicated, and I told you that it works, but if you were at different high schools, you'd have to memorize all of these intermediates and where it splits and the enzymes that facilitate these reactions, and I think it's ridiculous. You can always look this up. What you need to remember are the big picture items that we've established over here with respect to glycolysis. I've written up here saved, and I think you have been saved. Just focus at my level, but I do want you to read everything. Look at all the pictures in the textbook.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
Okay, take a moment, tell your neighbor everything you know about glycolysis, please. So I'm going to put the clicker down, and I want you to write down from memory now the reactants and the products of glycolysis. We're going to get this little kind of table going here so you can have it all summarized in one place. Try to do it on your own. So basically what I've done here is I've written down that we're going to make 2 pyruvates. From the breakdown of glucose, we reduce 2 NAD pluses to get our NADH products. And then we have 2 ADPs and 2 inorganic phosphates. That's just what the P little IP means. And then we have a net gain of 2 ATPs in the end. So you should be good with glycolysis. Ask me for help if you need to. You can go back if you need to. So if you keep going to 9.3 in your textbook, you're going to see this picture where there's a circle at the bottom and then there's a line going at the top. That line going at the top is this transition reaction. And then we have the citric acid cycle, or the Krebs cycle, named after the guy who figured it all out. Remember what the goal is here. The goal is to break this down and harness the energy that exists from the sunlight that was initially put into these bonds that share these electrons covalently between these atoms by chloroplasts that maybe occur in plants. And so sunlight's energy is sitting here. We need to harness that, literally break this and harness that energy by carrying it in the temporary form of these reduced NAD and FADH molecules and take those to the electron transport chain. So the whole rest of the goal here, this picture and what you see on this board, is take this and break it down. At the end of this, there will be nothing left. And that's okay. We don't want anything left. We want to harness as much energy out of this as possible. So let's find this together. Again, I would make someone read this, but let's read it together. At the bottom, figure 7-4, it says, now inside the mitochondrial matrix, or the middle of the mitochondria, each pyruvate molecule loses a carbon and attaches to coenzyme A, forming acetyl-CoA in preparation for the citric acid cycle. That's it. Let's just talk about the transition reaction. Let's go way up here to the top, way up there at the top, and I'm going to read that box. It says, in preparation for the citric acid cycle, the pyruvate loses 1 carbon via a carbon dioxide molecule and attaches to a molecule of coenzyme A, forming acetyl-CoA. So you can take a look at that picture there. Let me show you what it looks like in your textbook. So, of course, now they tell us that the pyruvate, which is over there on the left side of this picture, is C3H3O3, which I don't understand why it is, but okay, great. So we have this pyruvate. If oxygen is present in these higher organisms that have mitochondria, then this thing gets shuttled through a channel protein into the middle of the mitochondria, and we need to break this down. The first thing that happens is that a CO2 is broken off this thing. So let's imagine that we have a carbon, which is one of these black guys, and then 2 oxygen. So I'm going to take a blue and a blue. Those are oxygen atoms. And I'm going to, if I had time, I could put these together. So CO2, right? So this is a gas now. It's an exhalant that diffuses into the bloodstream, goes to our lungs, and we breathe it out. So that thing's gone. So really, this is what we have left. C2H3O. That's what I think that we should have left. Well, you can see up there at step number 2 that an NAD comes, and how many hydrogen atoms is the NAD going to grab? 2 of them, right? So let's tear off 2 of these. So another NAD gets reduced, sweet, and then something called coenzyme A comes in. I just have it right here on this little note card. Let's imagine that this then attaches to this. So this is acetyl-CoA. There's a problem. Does anyone see what the problem is? Well, this should be C2O and then 1H. But if you look at the picture, how many, I don't understand, I just don't get it. If you look at the left side of this picture on your screen, pyruvate is CH3. Then an NAD comes in and grabs 2 hydrogen atoms. But then if you look at acetyl-CoA way over there on the right side, how many hydrogen atoms does acetyl-CoA have? Isn't that awesome? Yes. It's like it gets reduced, NAD gets reduced, but it doesn't really take anything as far as hydrogen. I don't get it. Okay, whatever. So just imagine that this is correct, or imagine that the picture in the book is correct. I don't care. Just know that whatever is left at the end of the link or transition reaction binds to acetyl-CoA, and it then is going to go into the citric acid cycle. You don't need to know the numbers of atoms because there's obviously a discrepancy here. I'm the one who's probably wrong. I'm often wrong. It says before the citric acid cycle begins, pyruvate is converted to acetyl-CoA in the link reaction. Go ahead and turn to your neighbor and tell them what you know about the link reaction, please. Sorry about clicking around there. What I'm trying to understand in my brain is when we should do this. So let's do it right now, okay? So go ahead and try to write down, press pause, try to write down before me what is going to be the reactant, a reactant, and what is a product, or many different products of the link reaction. Okay, so this is what we have as far as the reactants and products of the link reaction. Let's just singularize everything. Don't worry about the 2 pyruvates coming out of glycolysis. Just let's focus on 1 because that's what your book does, and we'll talk about how it's duplicated in the end. Focus on 1 pyruvate. It gets bound to a coenzyme A, so a product is acetyl-CoA. You have a carbon dioxide molecule liberated, 1 for each pyruvate molecule, and then you have an NAD+, which is reduced to NADH. This happens in the matrix of the mitochondria. Why does it happen? It happens to prepare the pyruvate for the citric acid cycle, where it's eventually going to be broken down into nothing left. So that's the where, the why, and the reactants and the products for the transition reaction. But let's talk about the citric acid cycle. Let's finish reading here down at the bottom of this page. I'm midway through the paragraph on figure 7-4. It says, in the citric acid cycle within the mitochondrial matrix, acetyl-CoA combines with the oxaloacetic acid to form the 6-carbon citric acid from which the cycle gets its name. Citric acid is degraded in a series of redox reactions, finally producing oxaloacetic acid, 2 ATP, 6 NADH, and 2 FADH molecules. The oxaloacetic acid is now ready to bind to another acetyl-CoA molecule. The NADH and FADH molecules next move to the electron transport chain where additional ATP are produced. So that's a lot right there. So let's break this down and let's go, let's follow these cartoon boxes around this picture. It says way up in the top right. It says, the 2-carbon acetyl-CoA is combined with a 4-carbon compound, oxaloacetic acid, to form the 6-carbon citrate, citric acid. I think this is important. So what you need to understand is at the beginning of the citric acid cycle, this thing, acetyl-CoA, which comes out of the link reaction, binds to oxaloacetic acid. You can abbreviate this as OAA. This is something that is always there at the beginning of every citric acid cycle, and it's always there at the end. So it needs to be reproduced at the end of the citric acid cycle. That's how the citric acid cycle gets its name, when these two things come together and you get this citric acid that's over here on the right-hand side. You have a 6-carbon compound, but not for long. At least two carbons are going to be snapped off of that thing and then bound to oxygen atoms and exhaled as CO2. And where you're going to get those carbons from is this kind of this acetyl-CoA that came out of the transition reaction. Let's read this other box. It says, the reaction releases enough energy to attach a molecule of phosphate to ADP, thus creating a molecule of ATP. This is part of that substrate level phosphorylation. Something I would circle on your handout is that the GTP is the intermediate here, guanine triphosphate. I don't know why, but it's just kind of an intermediate before the ATP is made. Bottom left corner, it says, in several redox reactions, hydrogens are released and captured by the energy intermediates NAD plus and FAD plus. So we'll get through this detail right now. You can see it here. If we follow this around the circle, you can see that the 2 carbons that once existed here are going to be snapped off of this and exhaled as CO2. You can see that at about 3 o'clock and again about 5 o'clock, where the CO2 comes off of that kind of circle wheel. I guess let's just keep going. At the bottom, you can see that the coenzyme A is going to come off, and where do you think this is going to be recycled to? Probably back to the other part of the inside of the mitochondrial matrix where the link reaction is taking place. That's going to be recycled back to bind to another pyruvate molecule that's being broken down. And then you can see all around the whole circle NADs that are being reduced and 1 FAD that's being reduced. Well, press pause, and before I do this, why don't you try to figure out the reactants and products of the citric acid. Okay. So remember, what's important here? Where does it happen? Why does it happen? And the reactants and the products. If you were to summarize anything at the end of this, that's what's important. I think I've summarized that okay here. I don't really know the chemistry behind the FAD reduction, but this is the way that it comes out at the end, and the H2 and probably needs even more hydrogen atoms than even what we have in our DART, or lack of hydrogen atoms on that acetyl-CoA. But whatever, I'm not going to worry about it. Just understand that one of these is reduced here. We get 1 ATP made, a couple CO2s are released. I think this is a pretty good summation of it. What I'm trying to tell you on this slide is what happened here was we basically, when we rolled into the transition reaction in the Krebs cycle, we just focus on 1 pyruvate. But you have to remember that there's 2 pyruvates made at the end of glycolysis, so that all of this stuff happens twice. So on the exam, I might say, in the breakdown of an entire glucose molecule, how many FADs are made. And so you'll have to double this. You'll have to say in your head, okay, I'm going to double this because there's not just one pyruvate, but there's two. See that way over here in the cartoon that everything is doubled, that there's two terms of the citric acid cycle in the breaking down of these pyruvates.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondria. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Matrix of the mitochondria of all the cells is a cycle of reactions that will have Krebs and Nobel Prize. The cycle takes the energy and food and makes it into other forms your cells can use. Krebs cycle makes electron carrier NADH, which later brings electrons to the electron transport chain. When Krebs makes FADH2, its function is the same. Krebs also makes an ATP, another plankton thing, we're talking crabs. It's a citric acid cycle crabs. Carboxylic acid cycle Krebs cycle makes one ATP, 3 NADH, 1 FADH2. Right before the cycle's a transitional part, it's Krebs.
Okay, you can look at this on your own. Take a pause for a second and really try to understand this. Talk it out loud. I really think pointing at the picture, going over what's happening, really think that will go a long way to helping you. We're done for the day. We'll clean all this up by taking all these electrons that we've now grabbed with the NAD and FAD molecules, and we're going to convert them to ATP with oxidative phosphorylation the next time I see you. Thank you.